So let's start by reviewing mitosis. So I'm going to zoom into this first circle here, which is going to represent a cell that's in the G1 phase of interphase. And the chromosomes in the cell are going to look something like this. So we have paired chromosomes, which means that we have homologous chromosomes present. Now, in this drawing, I'm only drawing 2 pairs of chromosomes, or 4 total chromosomes, but in humans, we would actually have 23 pairs of chromosomes. So we can go over here and do some accounting. So for the number of pairs of chromosomes for a human, we would have 23. This would give us 46 total chromosomes because 23 times 2 equals 46. We also have a total of 46 chromatids, and that's because each chromosome only consists of 1 chromatid at this point. Now, because we have paired chromosomes, this means that we have homologous chromosomes present. And whenever we see that we have paired homologous chromosomes, it means that the cell is diploid. So now let's take a look at what's happening at this arrow here. So let's see what happens between this first circle and this second circle. So this arrow here stands for the S phase of interphase. And remember that the key thing that happens during the S phase is DNA is replicated. So DNA replication occurs during the S phase of interphase. And what this means is that the chromosomes are now going to look like the letter X. So we now have these X-shaped chromosomes instead of just having these bar-shaped chromosomes. And that's because each chromosome has now made a copy of itself that is attached to the original strand. So let's take a look at how many chromosomes and chromatids are now present at this stage of the cell cycle. So once again, if we're talking about humans, the number of pairs of chromosomes that are present at this point would be still just 23 pairs. We haven't added any new chromosomes, we've only added extra chromatids. So we still have a total of 46 chromosomes. However, the main kind of key difference at this point is we now have 92 total chromatids. And that's because each chromosome is now made up of 2 sister chromatids that are attached to each other. Now remember, we still have pairs present, which means we still have homologous chromosomes. And if we have paired homologous chromosomes, it means that the cell is diploid. So now let's take a look at what's happening during these arrows here. So these arrows represent mitosis. And remember that mitosis is made up of those subphases that we can remember using the mnemonic PP-MAT-C. And the key thing that happens during mitosis is that during metaphase, the chromosomes are going to line up along the middle of the cell like this. So during metaphase, the chromosomes line up in the middle of the cell. So what happens during anaphase is that the sister chromatids are going to get separated, and one sister chromatid is going to go to either side of the cell. So the key event that happens during anaphase of mitosis is that the sister chromatids separate. So what we end up with at the end of mitosis is cells that look kind of like this. Now you may notice that these cells are identical to each other, so they have the same genetic information as each other, and if you compare them to the parent cell, they are also genetically identical to the parent cell. So let's take a look at what we end up with at the end of mitosis. Now, first of all, it's important to note that we end up with two identical daughter cells. And each cell has the following number of chromosome pairs, total chromosomes, chromatids, etc. So each one of these daughter cells is going to end up with 23 pairs of chromosomes, which equals 46 total chromosomes. So remember, pairs are 2, so we have 46 total chromosomes. Now during mitosis, we split the number of chromatids in half, so each cell is going to end up with 46 total chromatids. And that's because each chromosome is now made up of only one chromatid. Now we still have paired chromosomes present, which means that we have homologous chromosomes, which means that the daughter cells are diploid. So these cells are diploid. So now let's take a look at what's happening during meiosis. So this first circle is showing us what the cell looks like during G1 of interphase. And the chromosomes are going to look exactly like the chromosomes that we talked about when we reviewed mitosis. So once again, I'm only drawing 2 pairs of chromosomes, or 4 total chromosomes here, but remember in humans we would actually have 23 pairs of chromosomes. The number of pairs of chromosomes at this point is going to be 23. So we have 23 pairs of chromosomes, just like how we started with mitosis. This means that we have a total of 46 chromosomes, because 23 pairs means 23 times 2, which gives us 46. We also have 46 total chromatids, and that's because each chromosome is only made up of one chromatid at this point. Now this cell is diploid, and it's diploid because we still have paired chromosomes, which means we have homologous chromosomes present. So now let's take a look at what's happening during this arrow. So just like mitosis, we have the S phase of interphase. And remember that the key event that's happening during the S phase of interphase is DNA replication. So what this means is that after the S phase has occurred, we now have X-shaped chromosomes. And that's because each chromosome is now made up of 2 sister chromatids, or 2 copies of itself that are attached to one another. So if we look at the cell at this point in the cell cycle, we still have 23 pairs of chromosomes, which gives us 46 total chromosomes. So we have not added any chromosomes to the mix here. We've only added extra chromatids. So the key difference here is that we now have 92 chromatids present, and that's because each chromosome is now made up of 2 chromatids. Now the cell is still diploid, and it's diploid because we still have paired chromosomes, and those paired chromosomes are the homologous chromosomes. So up until this point, mitosis and meiosis are exactly the same. So now let's take a look at what's happening during this kind of second set of arrows here. And so the second set of arrows are our first round of meiosis. And so this is our meiosis 1. And remember that the steps of meiosis 1, we can remember them using the mnemonic of PP mat C. Now it's important to note that during meiosis 1, some really important things happen. So first of all, during prophase 1, remember that the homologous chromosomes line up during a process known as synapsis. So during synapsis, the homologous chromosomes pair up. And then genetic recombination happens between homologous chromosomes. And remember that genetic recombination is also known as crossing over. And what happens during genetic recombination is that homologous chromosomes exchange genetic material. So to help illustrate that, we would have our homologous chromosomes lining up or pairing up with each other during synapsis. And then what happens is that the homologous chromosomes are essentially going to kind of switch arms with each other. So what we end up with are chromosomes that might look kind of like this. And recall that the purpose of this is to increase genetic diversity. Also recall that during metaphase 1, the homologous chromosomes are going to line up in the middle of the cell, and then during anaphase, the homologous chromosomes are going to be separated. And so one homologous chromosome from each pair is going to go into one of the new daughter cells, and the other homologous chromosome is going to go into the other new daughter cell. Same thing with the second group of chromosomes here. Just one of them is going to go into one of the new daughter cells. The other one's going to go into the other new daughter cell. So recall that during metaphase 1, we have something called independent assortment happening. So I mentioned that during metaphase, the homologous chromosomes kind of line up in the middle of the cell. Now, the order that these chromosomes line up in the cell is random. So they could line up like how we have it shown here, or they could line up in a different configuration. And so we call this independent assortment. Which just means that the side that each of these homologous chromosomes lines up on is random. And the reason why this is important is because it helps to increase genetic diversity. That's because in our first scenario up here, during anaphase 1, when these homologous chromosomes separate, they're going to separate in this configuration. But in option 2, our daughter cells are going to get a different combination of chromosomes because the chromosomes are going to separate in this configuration. So if we go back and look at these cells that exist at the end of meiosis 1, what we end up with are these separated homologous chromosomes. And we could end up with something that looks kind of like this. Or alternatively, depending on how these homologous chromosomes lined up during independent assortment, we could end up with an alternative of something that looks like this. So you can see that in our daughter cells after the first round of meiosis, we have a lot of different genetic combinations that can occur. Because of crossing over, we now have sister chromatids that are no longer identical. And depending on how the homologous chromosomes pair up during independent assortment, we can end up with a different combination of homologous chromosomes in each of our daughter cells. So let's take a look at what we have at the end of meiosis 1. Now, first of all, for the number of chromosome pairs, remember that we separated our homologous chromosomes, so we actually no longer have paired chromosomes. So each of these cells now has a total of 23 chromosomes. And that's because we took the 46 chromosomes that we had at the beginning and we separated those in two. Now, we still have 46 total chromatids, and that's because each chromosome is made up of 2 chromatids. Now, because we no longer have these paired chromosomes, it means that we no longer have homologous chromosomes, which means that the cell is now haploid. So now let's take a look what's happening in this second set of arrows here. These second set of arrows represent meiosis 2. And meiosis 2 goes through those same kind of subphases of PP, MAT-C. Now, the key thing that happens during meiosis 2 is that these sister chromatids are going to be separated. That means that during metaphase 2, the chromosomes are going to line up along the middle of the cell, kind of like this. So that during anaphase, the sister chromatids are going to get separated from each other, and each of those sister chromatids is going to go into a different daughter cell. So let's take a look at what we end up with regards to chromosomes in our final daughter cells. Now, I drew these chromosomes based on the chromosomes that we drew in the circles at the end of meiosis 1. But remember that if our chromosomes had independently assorted in a different way, we would have ended up with a different combination of chromosomes in the daughter cells. So let's take a look at what we end up with at the end of meiosis. So after meiosis 1 and 2 have occurred. So first of all, we end up with 4 daughter cells. And the key thing is that these 4 daughter cells are not identical to each other. They are also not identical to the parent cell because we kind of shuffled up the chromosomes in order to increase genetic diversity to kind of rearrange the genes that were on those chromosomes. Now, we no longer have those chromosome pairs because remember, we got rid of those during meiosis 1. So that also means that we no longer have homologous chromosomes. So each cell now has a total of 23 chromosomes. And those 23 chromosomes are represented as 23 chromatids. And that's because each chromosome is made up of one chromatid. Now, because these cells do not have paired chromosomes, which means that they do not have homologous chromosomes, these cells are haploid.